Hi everyone, welcome back to the channel. Today, we're diving into a topic that plays a huge role in pharmaceuticals and biotechnology, but often doesn't get the spotlight it deserves. Secondary Reference Standard Qualification. Sounds technical, right? Don't worry, I'll walk you through it step by step, and by the end, you'll know why this process is absolutely critical for drug development, regulatory compliance, and ultimately, patient safety. Let's start with the basics. Imagine you're a scientist in a pharmaceutical lab. Every test you run on a drug, whether it's purity, potency, or stability, depends on having a reliable reference. That's where reference standards come in. The primary standard is like the gold standard, rare, expensive, and sometimes limited. To keep testing practical, labs prepare secondary standards, but here's the catch. These secondary standards must be proven to be equivalent to the primary ones. That's what Secondary Reference Standard Qualification is all about. And if it's not done properly, results could be inaccurate, compliance could be compromised, and regulators won't be happy. That's why labs like ResolveMass put so much effort into making sure this process is rock solid. Now, why does this qualification really matter? Think of it this way. If your testing materials are unreliable, every result you generate becomes questionable. That's a big risk, both scientifically and financially. Qualification ensures that every test you run is consistent and compliant with global standards. It protects patients, supports large-scale projects where primary standards are limited, and builds confidence in the entire testing process. In short, it's not just a box to tick for regulators, it's the foundation for trustworthy science. Here's where the regulators step in. The International Council for Harmonization, better known as ICH, has issued very specific guidelines. ICH Q2 focuses on validating analytical methods. ICH Q6B is all about biotechnological products and their specifications. And ICH Q7A emphasizes good manufacturing practices for APIs and reference standards. Together, these guidelines act like a global playbook. If you want your results to be accepted worldwide, you need to play by these rules. Okay, so how does the qualification process actually work? Let's break it down. Step 1 is sourcing. You need to start with high-quality material from GMP-compliant suppliers who can provide full traceability. Step 2 is identity confirmation. This usually involves techniques like NMR or mass spectrometry to ensure the standard really is what it claims to be. Step 3, purity and impurity assessment. Here, methods like HPLC and qNMR help confirm the material's purity and identify any hidden impurities. Step 4, stability studies. Standards must be stable over time, so tests are done under controlled conditions to confirm shelf life. And finally, documentation. Without proper records, everything else falls apart. Detailed reports and certificates of analysis are absolutely essential. Now, of course, it's not always smooth sailing. Labs often run into issues like unexpected impurities, degradation during storage, or incomplete documentation. And if you're working with complex molecules like peptides or biologics, things can get even trickier. The good news? With proper planning, advanced analytical tools, and strong documentation, most of these challenges can be managed effectively. Here are some of the heavy hitters in this process. qNMR for absolute purity. HPLC for purity profiling and degradation checks. NMR for structural confirmation, LC-MS/MS for impurity detection, and of course, stability studies to predict shelf life. Each of these techniques gives a different piece of the puzzle. Put them all together and you get a complete picture of your reference standards quality. At ResolveMass, our team specialises in this exact process. We've worked with everything from simple, small molecules to complex biologics. Our expertise includes impurity characterization, isolation of unknown impurities, and developing reference standards from scratch. What sets us apart is transparency. Every result, every method, fully documented and validated. That's how we help companies not just meet compliance requirements, but also build confidence with regulators. So, let's wrap this up. Secondary reference standard qualification might sound like a technical detail, but in reality, it's a critical piece of the drug development puzzle. By following ICH Q2, Q6B, and Q7A guidelines, companies can ensure accuracy, compliance, and international acceptance. Done right, it speeds up approvals, reduces costs, and strengthens data integrity. Done wrong, well, the risks are high. That's why partnering with experts makes all the difference. If you're working on drug development and want to qualify your secondary standards with complete confidence, ResolveMass Laboratories is here to help. You can reach us through our website or drop us an email, and our team will guide you through a seamless, compliant, and reliable qualification process. Thanks for watching. If you found this video useful, don't forget to like, share, and subscribe for more insights into pharmaceutical sciences.